everyone. Today we are going to discuss the continuation of the last part, the last module in immunology. So today we are going to discuss about that, the third factor of innate immune system, that is the humoral factors. The humoral factors, it is known to be that one of the important factors which is contributed in both innate and acquired immune systems. So the major point in the humoral factor is the complement system. The complement system is known to be as C system. So in this C system, what we are going to be look, the first part is that MAC, that known to be a membrane attack complex. So when the complex of complement system, it is attaching with the any membrane of any organisms which is known to be a foreign particles. So in this case, in the MAC, if when the MAC is forming, so that maybe it is formed from that the major part of the immune system, the immune cell, macrophage. The macrophage is majorly playing the major role in the membrane attack complex. So what it is playing exactly, we will come to the next part of slides, that means the future slides. So the complement system 3, 4, 5A, the A part of that, and then remaining it will be created the chemotoxics. The chemotoxics will be directly involved into the production of the MAC complex. So how it is protected, that is the earlier module what we discussed in the phagocytosis, based on the phagocytosis. So the phagocytosis action, the particular organism was engulfed by the phagocytosis, and that place it is releasing the chemotoxics. So the chemotoxics is doing in the job of opsonization. That opsonization itself, it is creating the anaphylotoxins using these particular components of complement system 4, 5 and 3. So how it will work in these 3, 4, 5, the complement system, particularly in the A part, in the blood capillary, where the mast cell is playing a major role, where the histamines are released into that particular system when the infection was happened in that particular blood vessels. So the antibody was created, and then antigen presenting cells was founded, and then the histamines will be covered in this part. So coming to detail in the humoral factor, we may come across with the 3 different pathways. We will come with these 3 pathways. That is, one is the classical pathway, it is exclusively for adaptive immune system. Then the remaining 2 pathways, that is the alternate and monos binding lightning pathway, that is coming under the innate immune system. So we will go one by one. So these all the things is happening in the blood vessels. So the blood vessels itself, when the foreign particles is entering to our body, from that place, so the antigen, particularly where the antigen is known as a foreign particle, maybe the microorganism, either bacteria or virus, so the antibody is going to be attaching the place in the surface. That surface is maybe made up of by any type of protein, maybe polysaccharides. So in this case, what it is going to be doing in this complement system? The complement system having the different types. In the first one is the complement 1, that is C1. The C1 having 3 different parts. That is, the one is the Q, R, S, all 3 parts. The Q is the major core part, and then R is that the ball shaped part, and then S is the tentacles part. That is, the 6 different tentacles are present in this S part. So C having the 3 parts, one is that Q, another one is R, another one is S. So in this tentacle part is useful for binding with that antibody, which is not possible to remove from that antigen until unless it will be digested. So this attachment is happening when the antibody and then C1, the attachment is happening due to the usage of immunoglobulin M and immunoglobulin G. Then followed by that C1 attachment, then it will call for that C4 complement system 4. That's our complement system 4 itself having the 2 different parts. That one is that A, another one is the C4B. The C4A is where once it will be coming to attach with the microorganism, then the C4A will be released from that C4B, then only the C4B will be attaching that particular microorganism. That C4A is separated and then it will be holding in the blood capillary. Similarly, once the C4B is attached and then it will call for C2B, that C2 is called through the enzyme known to be a C2 convertase. That enzyme is created, then once the enzyme is created, then the C2 will be entered to that pathway. That pathway, then C2 will also having two parts, that is the C2A and C2B. Then similarly with the C4, so it will be also come and then joined with that that means C4B and then C2B. Then C2A is similarly with that present in the blood vessels. Then these two, both C4B and then C2B is joined together and then make that new enzyme, that is the C3 convertase, that will be called for C3. The C3 molecule also will come, it is also having the two part A and B. Then similarly the C3B is attached with that B part and then A will be separated. These all three is joined together and then create the new enzyme that is known to be C5 convertase. Here it is the major part. Once the C5 is before entering to the C5, the C3B is divided two parts. That one is attached with that C4B and then C2B. That another one will be directly attached with that the microorganism or any type of pathogen which is entered to our body. So why it is happening, we will discuss in that next part, that means the next module. So followed by this, then C5 will be called using that C5 convertase enzyme. Then C5 convertase enzyme is calling for C5. It is also two parts, that may be C5A, C5B. But here it is that exactly the C5B is not going to be attached with that C4, C2, C3Bs. So it will be attached just opposite to that, where the C3B is attached separately into the microorganism. Why it is attaching? Because this C5B is going to be making that new complex that is going to be a MAC, that is a membrane attack complex. So after the attachment, immediate attachment of C5B, then it will call for all other component which is present in the complement system, C6, C7, C8, Cn and C9. So then it will be making that new complex that will be very useful for us in the meaning of the complex material, that is known to be a MAC complex. So coming to that, this slide is a zoomed part of that, wow, what it is happened and then why this is very important. Here you see that the complex MAC complex, that is formed by C5B and C6, C7, C8, C9. So it is made that one complex. What is the duty of this MAC complex? It will be very important. Before I am going to that part, and then I will just show that earlier I said C3B is separately attached with that the particular microorganism, that is only for the reason of the phagocytosis action. So the C3B will be the binding material when the phagocytosis was happened. It was discussed in the earlier module of my talk. So coming to that next part, that MAC complex, what it is the duty of MAC complex, which is useful for the creating higher amount of sodium in our body with the water molecule, and then it will be enter in the high force to that particular microorganism or any type of foreign body to destroy that microorganism. This is the duty for MAC. So following this thing, that may be happen in any type of gram-positive or gram-negative bacteria, because it is having the lipoprotein or glycoprotein based cell walls. So this is very easy in that particular bacterial contaminations. So this is all the things is known to be a classical pathway. It is happening in the adaptive immune system. We must be understand the difference between adaptive and innate immune system. Innate means it is a natural immune system, but in the classical pathway is that adaptive immune system. So you need to be acquired any type of external resources for these particular particular pathway should be happening in our body. Followed by this, what is that alternate pathway? The alternate pathway is also the similar pattern. The only exception is that when the C3, the same way, once C complement system, all the things is coming together. So likely the same way, C4, C2, C3Bs are separated, and then C3 is already separated, divided in two, and then it is attached with that microorganism separately. From this place, only the small changes will happen. That is going to be a sandwich model. Why it is going to be a sandwich model? Because here C3B is divided into two. That one will be attached with the microorganism. Then it will call the new factor, that is going to be BB. So the BB also the factor, that factor having BA and BB. So BB is attached with C3B. Then it will be covered with that one factor, that is going to be a properdin, that is going to be a factor B. Properdin factor is useful for binding with that C3B and then BB. Then once again, that another one piece of C3B will be attached above that all the things. So it is looking like into the sandwich. So first layer in the down is the C3B, then BB, then upper one is the C3B. Then in this cutting part of C3B is happening due to the enzyme known to be a protease, that is also known to be a factor D. This is only the sandwich model happen, then the MAC complex will form. This is known to be a alternate pathway of particular innate immune system. Coming to that, the next pathway, that is the monos binding lactin pathway, that is MBL pathway. In this thing will be shown in after 2 days of the infection, then only we can identify or understand that what is happening. So in this monos-lactin pathway, both the classical pathway and alternate pathway, both the things will happen. But the changes is that it will be created that one monos binding lactin pathway. That means the monos binding point will be included in this pathway. So before I am going to that to explain, I will explain this thing. What is that active macrophage, and then what is the role of that active macrophage. When the active macrophage is started, then endogenic pyrogens, that is going to be interleukin 1 and interleukin 6, then the same way TNF-A. These all the things will be activated, and then in the macrophage itself, once the lysosome fusion, before the lysosome fusion, then the lysosome fusion was happened, then immediately automatically the antigen presenting cells will be reflected into that particular cells, then the MSC particles will take that one. So in this thing, where it will happen, the interleukin 6 will be activated from our liver. So it has the two phases of protein, acute protein phases. That one being the monos binding lactin pathway, another one is a complementary reactive protein pathway, CRP pathway. Here I will come to that first in the CRP pathway. What it will happen, the CRP pathway, the same thing. The cell will be affected by any type of microorganism. Then C1, C2, C3, 4, C5, and then C6, C7, C8, C9, it will be attached in the same way. MAP complexes will form. Everything is formed. But here the difference is that CRP pathway will involve with that CRP molecule. That is going to be a complement reactive protein molecule, which is the binding resource for acute, the particularly C1 material. So once it will be happen, then we will feel the high amount of fever or heat in our body. So coming to the monos binding lactin pathway, in this also we cannot find that it having the two different types. It may be so the immediate reaction once it will be affected by microorganism. Otherwise, it will be so for another 2 to 3 days later when the infection was happened. So what it will be happen here is the important one is the bacterium or any type of virus is entered to our body or foreign particles entered to our body. If the particular microorganism having the external wall that may be created by any external wall by protein or polysaccharides, in that case the MBL molecule, that is a monos binding lactin molecules, which is attached with that external wall. Until that it will be not created to damage the particular organism. Here, then the mask one, that means mask one cell will be come and attached with the different epitopes. These will be directly involving to the next step of all the classical or alternate pathway. So this is the way the map will work. So I will conclude in this particular module from this point. Hope we will meet in the next module in the maximum with that the other type of immunological studies, particularly in responses. Thank you.